I'll wait for, we got it. Welcome back, Ellen and Kate. Hopefully some other people join. But in the meantime, I can get started on a lot of the housekeeping. Host disabled screen sharing. All right, well, hi everyone, and welcome to Precision Health Bootcamp for Functional Magnetic Resonance Imaging. This is part 2 of 2. If you weren't with us for the first one, my name is Dr. Alexander Weber, and I've been an assistant professor and assistant professor in the Department of Pediatrics at UBC, and a staff scientist at BC Children's Research since 2019, so about 3 years now. I'm what you call an early career researcher. My research focus is on applying state-of-the-art MRI methods and analyses to investigate neonatal brain health and development. So I'm taking over for Lynn Williams for these talks and tutorials. She's an incredible computational neuroscientist at BC Children's, and she had a bike accident, so she couldn't be here today. I've taken over for her. So today we're going to be going over 2 things: Freesurfer for anatomical surface-based analysis, and we're also going to talk about diffusion weighted imaging, diffusion tensor imaging. But I'm in no way an expert in the field, so I have a couple papers published that use DTI, but I would say there are other people who have more experience. People like Stephen Miller, Tim Oberlander, Ruth Grunow, Jill Zwicker are examples that I can think of from BC Children's. Gotta turn it on. So this is kind of the overview of what we're going to do today. We're going to have more lecture, another tutorial, and then lectures again, and then another tutorial. So the first lecture is going to be about T1 and T2 weighting. So we'll dive a little bit deeper into that. I'll talk about Freesurfer, and then we'll do a tutorial. And then I'll go back to lecture and cover diffusion weighted imaging, and then go back to Sockeye and do a tutorial on that. So I'd like to acknowledge that we are gathered today on the traditional, ancestral, and unceded territory of the Musqueam people. And also, these lectures and tutorials are being shared under the Creative Commons Share Alike 4.0 International. You're free to share, copy, redistribute in any medium or format, adapt, remix, transform, build upon the material for any purpose. And for more information, you can visit this link. So today I'm going to be talking about Freesurfer. This is represented in the picture at the top here. It's a program out of Harvard that does a lot of good anatomical segmentation and surface-based analysis. So at the top here, we have the example where we have some basic tissue segmentation on the left, white matter, gray matter, deep gray matter structures. And we have some surface-based segmentation on the right. So you have identification of like Brodmann areas, I believe. And below that image, we have some pre-processing and reconstruction steps in diffusion weighted imaging. If you feel like there's a lot there, that's because there is. Just like fMRI, it's a pretty complex subject. Not only is there a lot to do to clean up our data for pre-processing, but after that, you actually have quite a number of possibilities of what you want to do with that. You can do tractography. You can do diffusion tensor imaging, which is a specific model of diffusion weighted imaging. You can do things like NODI or other multi-shell analyses. So the world is your oyster. So for our learning objectives today, at the end of the session, I'm hoping that students will be able to describe the difference between T1 and T2 weighted imaging, how to run Freesurfer on Sockeye, describe why water diffusion in the brain can help us understand things like white matter structure, describe how MRIs are able to detect diffusion in different directions, and finally, how to run something like QSI prep to pre-process our diffusion-weighted imaging data on Sockeye. So in our last session, we covered T2-weighted imaging. So let's just recap that. So we discussed spin-spin exponential signal decay, how water is bright on T2-weighted images as the hydrogen atoms in watery tissue. They spin too fast to really be dephased much. And tissue that is more of a mix of water and tissue, like gray matter, is a little bit dephased. And then some spins that are connected or touching fatty tissue, white matter, tend to be spinning slow enough that they will get a lot of dephasing. And so in terms of tissue contrast, they're going to appear dark. So this is a picture of a T1 weighted image. So let's just get some, right. So T1 is a little bit, I mean, at least in terms of the tissue contrast, it's kind of the opposite. So personally for me, I always remember if an image is T1 weighted in the brain, at least, because white matter looks white, gray matter looks gray. So fatty tissue like white matter ends up with a lot of signal. That's why it's really bright. And then gray matter ends up looking gray because it doesn't have as much signal. And then water is oftentimes black or no signal at all. So let's dig deep into why T1 weighted images look like this.
A quick question.
Absolutely.
So in diseases that have cerebral demyelination,
Okay.
is that something you'd see in the T1 and T2 images?
Yes, very likely. They won't. They're not tissue specific. So I'm not 100% sure how that's going to end up looking, but I mean, definitely clinicians use T1, T2, and something called flare, which is T2, but water appears dark for reasons I won't go into today. But they will use that to look for things like edema and white matter loss and stuff like that. But if you do want to do something more where you are using a technique that's more specific for myelin, you might use something like myelin water imaging. Yeah. Okay, so let's return to looking at spins after we've flipped them into the transverse plane. Hopefully you remember this from last session. And this time, to make things simpler, we're going to ignore the T2 decay or the transverse magnetization, and we're only going to look at the longitudinal magnetization. So the opposite of what we did last time. So we're only looking at longitudinal magnetization, which is different and independent of transverse magnetization. So in this example on the left, we've we've applied this 90-degree pulse. And so now we have the same amount of spins that are pointing up and down. And so they've canceled each other out. We have no net longitudinal magnetization. And then through something called a spin lattice relaxation. And again, that term may be outdated at this point, but we're going to use it for teaching purposes today. Some of these spins will start to slowly change their energy levels, and eventually we are going to regain our net longitudinal magnetization. So we go from 0 and then we slowly regain it. So this T1 relaxation is also known as T1 or spin lattice or longitudinal relaxation. It measures this recovery caused by spin lattice interactions, which I'll talk about in a second. And so the relaxation looks like this, whereas T2 decay was you lose signal. In T1, we are gaining signal. Since we consider the net magnetization being flipped through 90 degrees by the radio frequency pulse, it's tempting to think of the relaxation processes as simply being reversed, with longitudinal magnetization slowly turning from or through 90 degrees back to its equilibrium position. This would explain the gradual loss of transverse magnetization and recovery of the longitudinal magnetization. However, it would also imply that T1 and T2 are equal, and we know that this is not the case. So to drive that point home, I'm putting them on the same time x-axis, and you can see that T2 decay happens much faster than T1 recovery, and so you shouldn't think of them as being just opposites of each other. So T2 dephasing happens very quickly. Transverse magnetization is 0 after only a few hundred milliseconds, whereas T1 relaxation is much slower, and it takes several seconds before longitudinal magnetization fully recovers. So T1 relaxation is the loss of this extra energy from the spin system to its surrounding environment, or the lattice, hence the spin lattice relaxation. This energy loss is unrecoverable and represents the transfer of heat, hence the origin of the T1 synonym, thermal relaxation. This energy must be transferred somewhere, and that somewhere is into nearby nuclei, atoms, molecules, and this is all done through collisions, rotations, electromagnetic interactions. So at the most basic level, T1 relaxation is simply an energy flow between spins and their external environment, whereas T2 was spins and spins. So in order to induce the transitions needed for T1 relaxations, the fluctuations have to be at the Larmor frequency. That is that, let's go back to that precessional frequency that hydrogens are spinning at inside the magnet. So protons with intermediate binding tumble at a Larmor frequency. And by intermediate, we're talking about kind of binded to like fatty tissue. So protons or water in the fatty tissue will be closer to the Larmor frequency. They are going to relax faster, which means we're going to get the signal from them first. So that's why that appears bright. And then spins inside water are not really interacting with anything at the Larmor frequency, so we almost won't get any of that signal back until a long time. And then the in-between is the gray matter. So that is hopefully helping to explain some of our tissue contrast here. Okay, so you might be wondering why I'm talking about T1 weighted imaging for this talk. And the reason is we typically will want to have a T1 weighted image. Both we saw it in our fMRI, where we wanted some good anatomical information. So in adults, T1 pretty much gives us our best tissue contrast. And so that's kind of our workhorse for that. And if we want to do something like good skull stripping or brain extraction, volumetric labeling, so segmenting the different tissue, and then also surface extraction. So this is where you get into like folding and more advanced volumetric stuff. Eventually, you can do some gyrus labeling. Yeah, so you're going to need T1. Interestingly, in infants, pretty much up to 6 months, it's kind of the opposite. T2 images tend to be much better for tissue contrast, and that all has to do with the amount of water and the way that their brains are developing. Okay, so I know that was a very quick summary of Freesurfer. Again, I just want to show everyone that I'm not super familiar with Freesurfer, but if you have questions or whatever, essentially you can Google Freesurfer. They have a wiki. I believe they do have a course that you can take, hands-on training maybe. Oh boy. I do believe there is an online course in here somewhere. But yeah, so they will have resources, but today, yeah, I'm going to show you how to run this on Sockeye. Okay, so I think I'm still logged in. I am. That's great. So let's return to our bids folder. We only have one subject in here. Actually, what I'll do, so last session I showed you how to download data from an online repository. So I'm going to show you after I downloaded it what that looks like. Okay, so you'll notice some folders that are familiar: code, derivatives. This derivatives 2 I made out of, I was testing something. And then it doesn't have the source data folder because they didn't give us their raw data, but they did give us their nifty converted data. So let's look inside one of those. Picked subject 47 here.
So you ran your tester script that you had before on this, it should pass.
If they did it correctly, but I don't know if there's any guarantee that that's the case. That's a good point.
This would be essentially at that stage, you just pull this.
Yeah, it should. It should be. I think if you're going to upload to an online repository, that would be the nice thing to do. I found for this one there was a couple steps I needed to do. So I'm actually going to show you. Okay. So here we see that inside the subject folder that has an anatomy folder, a DWI folder, an FMAP, which is going to be used to correct for susceptibility distortions in EPI T2 star weighted scans, which are both our functional and our diffusion. And it has some functional data here, but we're not going to look at that today. So inside the anatomy, they just have a nifty file for the T1. So there should be a JSON file here. So they haven't included it, but they have in the main folder included essentially a generic T1 weighted JSON. So for my purposes, I actually would have to end up, let's see, T1 JSON.
You're already in 47.
Yeah, I'm trying to copy it. I wanted to. Oh yeah. I want to name it like this. I don't know, but oh, I see. I was not in the right folder. Sorry about that. Right. So I would likely want to copy that that generic T1 and put it into each folder, each subject. So the way that they have uploaded this has created a little bit of work for us, but it's not too difficult to get around. And I think if we ran the bids validator, it would say, where's the JSON files? Okay, so in order to run Freesurfer, we are going to have to load a module. So here we have module load Compute Canada. We'll do the GCC, and then we'll load Freesurfer. And we can make a directory in our derivatives folder. I've already created it. So I've called it Freesurfer. This is where I'm going to put any analysis that Freesurfer does inside of. And Freesurfer has one kind of main program. It's called Recon All, and you can tell it what your input data is, what the subject is, and then where you want it to put that data. Now this flag all is essentially just going to tell Freesurfer to run all the reconstruction. So if I just run Recon All with no flags, no options, it's going to give me this manual, and it's going to help me to understand if there's any other options I want to run. So for all, perform all stages of cortical reconstruction. So let's say that's what I wanted to do. So I could essentially put all of this inside of a PBS file and run it if I want to do that. And then let's say I ran that, and let's look at what we get. So get this here. Oh boy. Let's go to subject 47. So there's a lot of folders here. I think most of the data is going to be an MRI. And yeah, I'm not super familiar with all of the outputs that it's giving us. For me personally, I've had to run Freesurfer because it's generally called by other programs. So I haven't played around too much with this. But if you are familiar with Freesurfer and you want to know how to run it on Sockeye, essentially this is what you do. I think I've also, so I've given an example here of how you would run this. What's it called? PBS? Like what would it?
PBS job script.
Oh, job script. So this is how I would write my job script for Freesurfer.
What's the resources that it actually uses?
I don't know. Yeah. So when I mean, I made these just kind of all, I just use the same ones each time. So I don't know what the best way to do it would be. And
you always run it and test the QStat.
Like if it just cancels?
Well, you can do QStat -XF on the job that's been run.
Oh. In terms of all the output files, you can go to the Freesurfer page and try to look them up. I feel like they don't actually give much information on these. Yeah, there must be a better resource out there, but I didn't find it in time. So, okay, so that's just a really quick lecture on T1, T2, and a little tutorial on Freesurfer. So let's go back to our lecture. And now we're going to talk about the last lecture portion of the day, and it's going to be about diffusion weighted imaging. So diffusion weighted imaging is an MRI technique based on the tissue water diffusion rate. So it allows for very sensitive probing of water movements within the architecture of tissues. Most people generally are interested in white matter and white matter tracks, but I think that there is more and more now, there are various kinds of ways that you can analyze DTI data that can give you some interesting information about both CSF and like gray matter brain cells. So, and this water movement is based on something called Brownian motion, which describes the random motion of the particles suspended in a medium. It's named after, I don't remember the guy, but he was a botanist, and he had pollen in some water, and he noticed that it was moving under the microscope. And so we described this in a paper, called it Brownian motion, this random movement of the pollen. And it wasn't until Einstein in 1913 or whatever, his incredible year, where he had 3 seminal papers, where one of the papers that he published was kind of showing that what was moving this pollen was water molecules. And it sounds weird today, but this was essentially the first proof that matter existed. Because there was a time where there were 2 camps. There was essentially people that just believed that the universe was energy, and then people who believe that no, there's matter. I can't really wrap my head around how that's a thing at the time. If you guys are familiar with Boltzmann, he was ridiculed for believing in matter, and essentially, which may have led to him hanging himself. So that's some interesting and fun science history for you. Okay, so let's get back to Brownian motion. So basically, if water is free to move around, it will do just that. It's going to follow this Brownian motion and just kind of wander randomly. Whereas if it's restricted, it's not going to be able to move in a certain direction, but will continue to diffuse and move in that direction where it's free. So these are the basic things that we need to understand. All right. So this picture is going to be complicated because I haven't gone too much into pulse sequences, but I'm going to try to kind of explain how we're able to measure water diffusion in specific directions. So the way in which diffusion information is extracted from the tissue is to first obtain a T2 star weighted image with no diffusion attenuation. So this is really easy. We've done this before, for example, in fMRI, and this is known as our B equals 0 image. So essentially just means that we haven't applied a gradient, a diffusion gradient. So next, the ease with which water can diffuse in a direction is assessed using gradients that are applied in very specific directions. So the simplest way to do this is to get 3 orthogonal gradient directions, so X, Y, Z. And so we're going to use this for the rest of the explanation. So how do these gradients work? We apply this strong symmetrical gradient in these various directions on either side of this 180-degree pulse. And the stronger these gradients, the more pronounced the diffusion-related signal attenuation is going to be. So stationary water molecules are going to acquire phase information from this first gradient. And after the 180-degree pulse, they're going to be exposed to the same gradient if they haven't moved, which undoes all the effects of the first, and hence we should retain full signal. So if water does not move, we should, in applying these gradients, we should retain a strong signal. However, moving water molecules, on the other hand, acquire phase information by the first gradient, but as they are moving, when they're exposed to the second gradient, they're not in the same location, and thus they're not exposed to precisely the same gradient after the 180-degree pulse. Hence, they are not rephased as they lose some of their signal. So the further away they are able to move, the less successfully they will be rephased, and the less signal will remain. So maybe the easiest way to think of this is this first gradient is sort of like, it almost tags our water molecules, and if they stay there, then we're going to see them again. And if they don't, then we're going to see less of them. That's my very simple way of explaining it. Okay, I can kind of show you how this looks. So here we have our simple 3 orthogonal directions. AP is anterior, posterior, so like this. LR is left and right, and superior, inferior is like this. So those are 3 perpendicular directions. So remember, if diffusion gradient is in a specific direction as applied, water that can move in that direction will show little signal, whereas water that is restricted in that direction will be bright. So let's just look at this picture on the left, or actually across all of them, we'll see that ventricles are dark because water is able to move freely. But inside our tissue, so for instance, on this left one, inside white matter, especially in this axial slice, these white matter fibers are pretty much in the direction of, let's say, forward and backwards. And so water is able to move in that direction. And so we get this signal dropout. Versus left to right, in that same area, we actually see brighter signal because in that direction, water is restricted. So I hope that gives a sort of a good visual impression of diffusion weighted imaging.
So if you had like a stroke and blood is no longer able to flow in one of those directions, will you see a blockage in that, like if you go back, like where you expect to see the dark, you start to see the white, that would mean that the water's blocked at that spot? Yeah,
I do think that, let's look this up. I do think it is very bright. Yeah.
Yeah. Okay.
So I guess with the stroke, you do end up getting this occlusion and diffusion imaging. It will turn up very bright. Versus I think T1 and T2 weighted images might not show that as clearly. So just from a clinical perspective, a simple diffusion weighted scan can be pretty powerful. Okay, yeah, go ahead.
Sorry,
No worries.
This kind of diffusion weighted imaging generally study the water whatever index, and then in brain we have blood which contains a lot of water, and also we have some CSF, some whatever liquid is also contains a lot of water. Then this DWI imaging has the ability to discriminate from water in blood and water in CSF.
Yeah, I think because CSF, there's going to be, or in vessels, there's going to be that perfusion. There should be a difference between them, but I'm not.
Oh, it's okay. It's okay.
Oh, it's okay. Yeah, no, I think you are onto something, but I'm not 100% sure how to answer it. So one example of the use of diffusion weighted imaging is something called DTI, so diffusion tensor imaging. And this allows for the indirect measurement of the degree of anisotropy, which is a fancy word for direction bias and structural orientation. So while diffusion weighted imaging refers to the contrast of the acquired images, DTI is a specific type of modeling of the diffusion weighted data sets. And there's many other models, and they're all fighting to be the right one and give specific information. So the basic concept behind DTI is that water molecules diffuse differently along tissues depending on its type, integrity, architecture, and presence of barriers, giving information about its orientation and quantitative anisotropy. Based on this assumption, one can model the diffusion process by an ellipsoid. So down here, bottom left, which can be mathematically represented by a 3x3 symmetric matrix, also known as a tensor. That's where the DTI gets its name. And essentially, you're going to have 3 primary eigenvectors and 3 primary eigenvalues. And so with this model, it's possible to create a fractional anisotropy map. So that's here in black and white, where fractional anisotropy is an index for the amount of diffusion asymmetry within a voxel defined in terms of its eigenvalues. So the value of FA varies between 0 and 1, so it can't be lower than 0 or higher than 1. For perfect isotropic diffusion, so that's like a sphere where water doesn't have a specific direction, FA is going to be 0. With progressive diffusion anisotropy, kind of down here, the eigenvalues become more unequal, the ellipsoid becomes more elongated, and the FA goes towards 1. Right, so on the FA map, brighter areas are more anisotropic than darker areas. So here in the white matter, we can see very bright. So that means very anisotropic, versus gray matter, it's more dark because those are more spheres and water is less restricted, at least in a specific direction. So you can also create something called a principal diffusion direction map, which is this beautiful, brightly colored image that are often used in posters to impress people. So this is essentially the same as an FA map, but with a little bit more information. So this map assigns colors to voxels based on a combination of anisotropy and direction. So before with FA, we couldn't tell which direction it was specifically limited to. So the color assignment is arbitrary, but in this case, we have Y as green, X as red, and Z, which is pointing out, as blue. So that gives us the color, and then the intensity is going to be based on the FA. So another example of what you can do with diffusion weighted imaging is something called fiber tracking. So it's a pretty simple concept once we understand the FA stuff. Axonal tracks are commonly mapped using something like a deterministic method. although there are other methods, probabilistic, and so on. But in a deterministic version, we can select a seed voxel, and then from there we can follow the primary eigenvectors outwards and see where they point, and then look at that next voxel and look at its primary eigenvector, and so on and so forth. And so what we end up with is these fiber tracks. Okay. So the thing is, diffusion weighted imaging comes with a lot of messy data, and we're going to try to clean it up. So that's what the first step is. We're going to have to pre-process our data, just like we did with fMRI. And so the artifacts in diffusion weighted imaging are mainly related to these gradient systems I introduced, but also Paul sequence acquisition strategy and motion, again, motion. So diffusion weighted imaging is generally collected over the entire brain with repeated acquisitions while varying the orientation or the magnitude of the diffusion gradients. DWI, like fMRI, has low signal to noise ratio, and the resolution is also pretty low, oftentimes pretty similar to fMRI, and very susceptible to motion. So to reduce the influence of motion artifacts, the scan time has to be reduced. And so once again, we meet our friend echoplanar imaging to try to speed things up. But this then introduces things like susceptibility-induced artifacts that we're going to have to correct for.
Do people look at the same kind of task-based, from rest to task, rest to task with DWI? See the kind of flow of like expected change in direction of flow in your colored map.
So conventionally not. However, I do believe there is such a thing as functional DTI. I forget what it's called. I don't know if anyone on the chat has an idea. Is it only us four? No, there's more people. Okay. I only saw 4 of us. There is definitely, I haven't done it. I'm not familiar with it.
Not as popular as fMRI.
Not yet. Oh, what is it called? I'll have to get back, but there is such a thing. And there's even like functional spectroscopy. Yeah, it's all pretty interesting. I mean, typically people don't think of DTI as kind of being necessarily like going to be changed by a task because it's thought of as more structural. But I do think there is some people who, yeah, I'll have to get back to you. But
I just wonder if,
yeah,
if you'd ever see, like, instead of the flow of oxygen, if you'd ever see something as dramatic as like a constriction of a blood vessel.
Well, yeah,
to something, right? Like, because I think the DTI is showing you the flow of water. So if you see a disruption of flow in response to some task, as opposed to a change in the oxygenated blood in response to some task.
Yeah, I don't know how sensitive DTI is to blood vessels themselves, but you can look at blood vessels using like susceptibility weighted imaging to get like a quantitative susceptibility map. But you could also do arterial spin labeling, which will look at cerebral blood flow more specifically, which I think is what you're sort of getting at. And I was mentioning before that some people recommend functional arterial spin labeling over bold imaging. Yeah, there's a lot. So essentially, today I'm going to be going over these pre-processing steps. And again, there's so many different ways you can do it that if there's a standardized way to do it, it might be better for the field as a whole to sort of follow this correction, head motion correction, something called eddy current correction, which we didn't have in fMRI. We still have this susceptibility distortion we want to fix. We want to register it to a shared space, and we want to do data quality control. So let's exit this. Okay, so what I'm going to be showing you for the pipeline is something called QSI prep. I forget why it's called that. It's like Q space. I don't remember what Q space is, but that's what it's named after. But your data needs to be in BIDs again, and it's going to run a pre-processing pipeline that's going to use FSL, ANTS, MRI, tricks, other like diffusion Python, DSI Studio. So it uses a lot of other software in order to do its magic. Again, it can be run through a Docker container, a Singularity container, or you can install it on your computer using pip. I think. Oh, here are their external dependencies: ANTS, AFNI, Freesurfer, FSL, DSI Studio, MRI tricks. Okay, so let's show you how to do this.
So it also operates with ND files.
Yes, and again in BID format. So actually, let's look at what our files look like. Okay. So in subject 47 again, this is just the data we downloaded from the open repository. We can go inside the DWI folder. We have a DWI Nifty. I can show you what that looks like on my home computer. I downloaded this earlier so I can show people. Okay, so we're actually going to have interspersed, we're going to have those B=0 files, which are going to be brighter. So actually, as soon as I do that, we need to change this contrast again. Okay, so this is going to be fun. So maybe take a look here. And as we go through the different directions, we're seeing different diffusion restrictions, and so signal dropout, so on and so forth. And instead of 3 directions, this DTI was obtained in many directions, and I can show you what that looks like once we run our pre-processing.
So it's not just X, Y, Z, it's coming at every.
Yeah,
right?
we're doing a whole sphere, and like oftentimes people get 30 to 60 directions. And yeah, it's more recently that people are getting multiple B values. So instead of just B0 and B some strength, let's say 1,000, they're also requiring like B1000, B2000, stuff like this. I think actually we can find out what those strengths are here. So we have our JSON file, but now we also have this BVec and this BVal. So the B value vector and B values. So let's look what those look like. So these are our B value strength. So we have our first image is 0, then 995. I don't know why this person has these small changes in B value. I haven't personally done that. And vector should be the directions, so it's going to look crazy. So these are the directions. I mean, I'm going to visualize it in a second, so we'll understand. Okay, but anyways, we have our JSON, we have our BVal, BVec, we have our Nifty. This is ready for QSI prep to use. In order for us to be able to use it, we need to, we're going to again do what we did before. Module load Singularity. Okay. No, I think it's how do we return to? Sorry, there is something I have to load before I can load Singularity. I don't remember what it is.
I think you have it if you module this.
Okay. Oh, I think it's because I have the Compute Canada.
Yeah.
So remove?
Yeah. CVM.
There we go. So we can load Singularity, and then we're going to want to install this somewhere. So for me, that's my project space, installations. I made a folder QSI prep. So I would do like make directory QSI prep. And then I would do Singularity pull, put that installation directory, what I'm going to call the Singularity image, and where to get it from. This is the address. Oops. What happened? Right. So after I've installed the QSI Singularity image, I can then create my PBS, what's it called? JavaScript. So this stuff we've gone over before, and inside you need to load Singularity. Let's see if we usage. So again, just like FMI prep, there's a lot of options. I'm not going to be able to go over them all today. And hopefully it tries to explain what each option does. So in my case, let's see what options I gave it. I just told it which subject to do, gave it the Freesurfer license. The output resolution, I'm not sure why it requires you to input it. I just put what the actual resolution that it acquired it in. So I can show you kind of. If I load FSL, I don't remember which one I need. Come on. No. I guess you could make an alias that just does this really quickly for you. So let's go to my diffusion folder. I just want to show you, FSL has many subprograms. One of them, FSL info, can kind of just give you some basic information. So the resolution here is about 1.8 by 1.8 by 2. So when I run my QSI prep, I'm telling it to put into a 1.8 resolution. Okay, so I've written that here in my code, QSI.pbs prep. So this is what I ran, and I can show you the results. So I brought it over to my computer after it was done, because anything you want to do kind of with your GUI, you're going to need to do at your home computer. Maybe it's just easier for me to do this. So just like FMRI prep, it gives you this boilerplate, and it's going to tell you kind of what it did, and also give you images just for you to check how much you like it. A lot of this is the same anatomical stuff that we saw before. Some denoising, bias correction. So it does a before and after. here is where it shows you the directions that it acquired, and you do want kind of a whole sphere acquisition if you do acquire more than 3 directions. I think it messed up here, and the reason that you see this line is because this data that I acquired from this online repository, I don't know why they did this. But when they acquired this kind of field map correction, they they missed part of the brain. And so I think QSI prep, really messed up because of that. So this is exactly why you kind of want to look at your boilerplate and see whether or not what QSI prep did was good. And I would say in this case, that's not good. But I'm working with the data that I downloaded.
So is this how you would, I guess when it comes to reporting this kind of data in publications, some of those reports, do you ever include these HTML outputs or?
Oh, I haven't done that, but I mean, that's a great idea for supplementary. I mean, there's just so much that should be shared now that we have the ability to do that.
It's a pretty report too.
Oh, it's so pretty. And again, here's all the steps it does, and it tells you.
So this must come from the same group, QSI prep and MRI. It looks very much the same.
But it might be QSI prep sort of just copying the same. I don't know. I don't know if it's the same group, but you're right. It looks exactly the same. Oh, we're here. Do not imply that the authors of FMRI prep in any way endorse or support this code or its pipelines. I think they might be different people. But yeah, where does it say who made this?
10 BBL.
Yeah, I don't know. So let me just exit these. Okay, so believe it or not, that is pretty much it in terms of QSI prep. But I figure, we have another hour. I'll probably end early, but I should show you some other stuff that we can do now that we've pre-processed our data. So one of them will be just using this tensor model. We're gonna acquire, for instance, our primary vectors and our FA map. So if I were to do this in a PBS script, I would load FSL because I'm going to use an FSL program, and I'm going to use something called DTI fit, which hopefully I still have this module load here. DTI fit. So if I just type that out without any options, it should give me some information of sort of what it's doing. If you want a full user guide, FSL does a pretty good job kind of explaining what it does, how to use it, what options to use. So here I'm running DTI. Let's see what K is. K is the data. So I need to give it my pre-processed data from QSI prep. So here I've defined sort of that main folder, derivatives, QSI prep. And the O flag is output. So I'm telling it to put it in a new folder in derivatives called DTI and to name all the files DTI fit. M is a mask. So remember we were talking about brain masks. This is because you just want to look at the parts that are the brain, not anything outside of it. And then you're going to give it the bval and the bvec files. So let's pretend like I've run that. So these are the files I'm going to get. FA, MD is median diffusion or mean diffusion. V1, V2, V3 are the three eigenvectors. So I can show you what this data looks like now after it's run. And let's say I brought it to my computer at home. It is in a different orientation. You can fix that, and QSI Prep shows you how to do it. I'm not sure why they decided to have it in this orientation, but they must have their reasons. Okay, so this is an FA. So anything that's bright tends to be very restricted or anisotropic. Anything that's dark is, more like the CSF or things that don't have barriers or specifically direction. So if we load our primary eigenvector, we can modulate this one by FA. And so this is what I was showing before. Incredibly beautiful. So blue is kind of in the Z direction, up, down. I forget what, we're gonna find out in a second. I think red must be left, right. Let's get, oh geez. Yeah, you can kind of see left, right here. And green must be the other direction. Kind of forward, backwards. Okay, so that's how you would analyze your DTI data. So it was really simple. It was just one line of code, right? And you could obviously change this so it runs over multiple subjects. And because it's in BIDS format, it's going to be really easy to program that because they're written in a very standard way. So you would just really have to make this subject ID a variable that you would then go through a list of the subjects that you want to look do. Okay, I'm going to show you. Oh, I'm not going to show you this. Sorry. I tried to get this to work and it kept failing. So the thing with QSI Prep is it is mostly a pre-processing pipeline. However, they do have these reconstruction scripts included. So if I wanted my images to be in the orientation I'm used to with FSL, you can run reorient FSL standard. If I want to run a naughty pipeline, which again is this, it's a different model than a tensor model, and is used for multi-shell DTI to get kind of more specific architectural information. But I couldn't get it to work on Sockeye. So yeah, I'll have to investigate. I think it's a Python thing. I might have the error still.
We don't have to debug this live.
Oh, we should debug this live. Do it live.
Maybe it's not contained, like Python path error probably.
Yeah, so I just want to show how I would run a recon. Oh God, cat. So it's pretty similar to the way you originally run it, but now you would tell it, oh, I just want to do reconstruction. Where do I find the pre-processed data? It's where I told it to put it before. And then what do I want to run? And so again, that would be, you would pick from this list and you would just type exactly what it says here. And if you don't want to use Singularity, you could actually install Naughty yourself through a Python environment. So I'll show you that, but again, I failed to do that. That didn't work. So let's say you activate your Python environment. You could then install that Amico Naughty pipe or algorithm like this. And then this is what you would have to run as a script inside your PBS script. So essentially you just give it a bval, bvec. It creates a scheme, which is, I think it just combines them. And then, yeah, you just tell it what preprocessed data to gather and the brain mask, and then it will run everything. So I did run that on my home computer for illustrative purposes. I think. Oh wait, I think I put it here. Yes, there is. I'm not an expert on naughty, so I mean, this one is orientation dispersion. I feel like this has to do with fanning, but I can't remember. This one is going to be like CSF type information. Definitely very bright is kind of the ventricles. This one should be neurite dispersion, but again, I'm not an expert, so I can't. This actually looks really dirty. Okay.
Because it shows the importance of singularity in these complex toolkits.
Okay, last one. So we'll do tractography. This was actually really fun because I'd never done tractography before, so I had to learn it just for this little tutorial. This is the PBS script that I would write. It's essentially using, again, it's using this reconstruction. So I think I ended up using this one. And it uses MRI Tricks, which is another software package. So I show you what that script looks like. Again, I'm running singularity because I'm calling my QSI prep, and then I'm telling it to do this a reconstruction. The reconstruction I want it to do is MRI Tricks, single shell, blah, blah, blah. It requires for you to have run Freesurfer. So I think I had to have run that previously. And that's it. You would run that, and what you get I think are these files. And if you can see, the biggest file is 10 gigabytes, which is relatively big. And it is this .tck, which I think are all the tracks. And so what I ended up doing after I brought it over to my computer is I edited it because it might be too many fiber tracks to actually visually look at. So I reduced it to 200,000. And then I ran MRView, which I think is an MRI Tricks software package. Okay, so I'm not super familiar with this, so please be patient. Oh God. Okay, how do we get rid of image? View. So essentially what it's done, I wish I could figure out how to clip this image so that we could look inside, but I didn't figure that out in time. But yeah, these are essentially a full tractography of the brain, which looks really, really cool. And obviously you can do a lot more if you had a specific seed region and you wanted to see where the tracks are from there. And then if you had two groups and you were interested in a specific region, you could see how their tracks are different. But yeah.
So this figure is amazing, and my point is that it's a little bit complicated, and it's really hard for human eye to identify the difference. So we have any software, for example, compare the phase 1 and phase 2. What's the difference in the tractography in terms of different?
Yeah. So actually,
This matrix 3 do this.
I think it all depends on what you're doing. So for instance, a lot of, if you do like if you are looking at FA, so for DTI, if you were to try to compare the entire white matter, you would have such a huge, you have so many voxels that you're comparing, that you'd have an incredibly difficult multiple comparisons problem. So FSL has a program called TBSS, and it kind of, at least my understanding, this kind of breaks it down into a white matter skeleton, and then further analysis, you do try to yeah, here's the skeleton. So instead of the whole white matter, it's just kind of this green area. And then this is kind of what you would get. This would be like a difference between two groups within that skeleton. But for tractography, I'm not familiar with what you would run in order to compare groups and things like that. Oh, sorry, I'm not sharing the screen. Oh my goodness.
Okay, there's a question for you in the chat. Kate.
Earlier, I mentioned that T1 is suitable for imaging adult brains and T2 for infants because of the difference in water content. Would it be useful to perform both T1 and T2 and combine the info somehow? Or are any added benefits limited, making it not worthwhile? No, I think that's an excellent point. I think that, so for instance, I'm going to share the screen this time. I think that's not a naive question. I think that's an excellent question. 1 second. And it all really depends, again, on the age, because pretty much from birth to 6 months, you can, it'll look good on T2, but around 6 months, there's almost like no tissue contrast in any of the things you use. So it can get quite tricky. So 1 sec. Oh yeah, so so many of these pipelines are designed for adults. And then when you try to apply them in babies, it has a lot of assumptions that just won't work. And you'll just get, like for instance, brain extraction is really difficult in babies using any of the pipelines that were designed on adults.
The skull's not the single shape, right?
Oh yeah, that's fair too. Yeah, totally different shape. So I was using something from the Human Connectome Project people, and it's like a subgroup of theirs where they're doing the developing human connectome. Far cooler stuff than I could ever hope to do. So they have a freely available pipeline. I'm sure there's a way to get this running on Sockeye. This says Docker, but maybe there's a way to do it with Singularity. And so for instance, it does take in both T1 and T2 images. It is required to take a T2 image, but I think it will also take in a T1 image to try to improve all its fitting and assumptions and stuff.
Yeah, there was a bit on Singularity there.
Oh. Okay, maybe I spelled it wrong. So yeah, I mean, I think that's an example. I think there's also Freesurfer infant Freesurfer, which you could also try to run on your anatomical data. And let's see if we can find out. Oh, I'm not sharing again. Sorry. Share screen. So here, I just looked up infant Freesurfer. I'm not going to be able to do this live and figure out what it uses, but I imagine it uses T2, maybe T1.
T1 weighted neuroimaging.
T1. Oh, I think this is, oh yeah, interesting.
That's it.
Oh, but it also is from 0 to 2 years. And I was mentioning that T2 is kind of best for 0 to 6 months. So I'm not sure how well this would actually work below 6 months if it only wants T1. More questions? Oh, you're welcome. Any questions from here? Any more questions online? You don't need to apologize. I love your questions. You're doing a great job.
Thank you. Anyway, so now we know when the stem that the T1 and T2 are generally show the opposite signals because T1 has the brighter water and the frozen darker and the T2 show the opposite. But my question is that why do they derive this kind of opposite signals? Because if they are totally opposite, while we know T1 and already know T2.
So they're not. So they're so
not.
okay. That's a great question. So they're. I love that. So they're not actually opposites, and they really are kind of independent physical processes. And so because of that, they are. They will give you different information. So I think there's a really good. There's a really good. Picture and it shows, no, I'm not going to be able to find it.
Not brain.
That's okay.
T1 and T2 can also brain scan other part of the body.
Yeah, oh yeah, absolutely. Oh, I wish I saved the image. I'm so sorry. There's an excellent image and it does a really good comparison of how they are very different, like biophysical processes. I don't think we're gonna find it. No. Yeah, sorry. I wish I could show you that. I think it gives a better visual kind of picture of the differences. No problem. The only thing I would want to say is there are the kind of teacher evaluations or whatever. If anyone has the time, it really helps me both for improving and for putting in a teaching dossier, which eventually I have to show to some committees. So yeah.
Yeah, we'll put that in the chat.
In the Slack chat.
Yeah, Slack chat.
And is this the final session for our boot camp?
We have tomorrow.
We have tomorrow. What's happening tomorrow?
Deep lab cut. Which
Whoa.
tracks animal movements.
Oh boy.
Videos.
Okay, well that's exciting.
And then we have a party, and everyone should come to the party at BC Children's tomorrow afternoon.
Tomorrow, 4 o'clock, BC Children's. The information's in the Slack channel, probably.
Yeah.
Okay. Okay. If there's no more questions, I'm gonna call it.
Awesome.
Okay.